In the previous session, I discussed about estimation of gene order and map distances to construct linkage map using cross-breeding technique. Here, I would discuss about the concept of creating linkage map using pedigree analysis and LOD score. How to create linkage map with different types of organisms. There are three different types of situations. First, in organisms like flies and mice, an experiment can be designed and planned using true breeding types, heterozygous genotypes, etc. In such cases, the linkage map can be created using the method I discussed in my last session. Next, there are organisms where experiments cannot be designed, example in humans. In such cases, we utilize the pedigree data to make the linkage map. The third category of organisms like bacteria where meiosis does not occur. That is, there is no crossing over to calculate the recombinant frequency. In such cases, we use interrupted mating experiments for mapping the genes. In case of humans, to create linkage map, the geneticists rely on pedigree data, which are often incomplete, and calculate the LOD score, that is, logarithms of odds. It is a statistical method to estimate whether the two genes are located near to each other or not. The principle here too is linkage disequilibrium. That is, the genes that are close to each other or linked are inherited together. As LOD is a statistical tool to calculate the likelihood of odd ratio, first a hypothesis needs to be put forward. So first, a null hypothesis H naught is put forward stating that no linkage occurs between the two genes. That is, in that case, the recombinant frequency theta is equal to 0.5. Remember, if recombinant frequency is 0.5 or above, the two genes are not linked. Then an alternative hypothesis HA is made stating that linkage occurs between two genes. In such case, theta is less than 0.5. Then the LOD score is the logarithm of ratio of probability of birth sequences with a given linkage value to the probability of birth sequence with no linkage value. Steps to calculate LOD score. First, study the family history for the particular case and draw the pedigree to identify the affected and the unaffected individuals. Then determine the mode of inheritance. Second, cross out the individuals whose information is incomplete. Third, among the individuals for which the information is complete, determine as to whether they are recombinants or non-recombinants. If the genotype is consistent with the parent haplotype, They are non-recombinants. If not consistent with the parental haplotypes, they are recombinants. You should remember that even an unaffected individual can be a recombinant. Next, count the number of recombinants and non-recombinants. There are three ways to calculate the LOD score depending upon the data we get. First instance, when the recombinant frequency theta is provided, we would directly go with the equation given below to calculate the LOD score. If the distance between the gene marker is given, we divide the value by 100 to get the recombination frequency theta. Because recombination frequency percentage is actually the map distance between the genes. Third instance, if we need to calculate the recombinant frequency, then use the equation below with theta values 0.001, 0.10, 0.15, 0.20, 0.25, 0.30, 0.35, 0.40, 0.45 and 0.50. The correct theta value would yield the highest LOD score. You will understand it better when we do it with an example. If the LOD score is less than 2, it means that the two genes that we are considering are not linked to each other. If it is between 2 and 3, the result is inconclusive. If the LOD score between the genes is greater than 3, it shows that these two genes are strongly linked to each other. That is, the likelihood of linkage occurring at this distance is 1000 times greater than no linkage. We take it 1000 times because it's the log value. 3 is the log of 1000. To understand LOD score estimation, we'll go with an example. Now, here a pedigree is shown with linkage between nail patella syndrome and blood type A, B, AB, and O. We need to statistically prove by LOD score whether these two are linked or not. The shaded individuals here are the affected. The first thing that we need to know is the pattern of inheritance. Here, the mode of inheritance of the syndrome is seen to be autosomal dominant, as the syndrome is present in each generation irrespective of gender. It can also be seen that the dominant allele for the syndrome is coupled with A-type blood group, except for one individual, that is third generation individual 5. Therefore, this individual can be considered as a recombinant type. So, when we calculate the recombinant frequency out of 8 progenies in the third generation, one is a recombinant. So the recombinant frequency is 1 by 8, which is equal to 0.125. The map distance between the two genes, that is the Neil-Patau syndrome gene and blood type, is 0.125 into 100, that is 12.5 centimorgan. Now here, since we know the recombination frequency, that is theta is 0.125, we can go for estimation of LOD score by using the formula for LOD. Since the theta value or the recombinant frequency is known, that is 0.125, we can directly go for calculating the LOD score. Now here Z is equal to log 10 by ratio of theta. Theta here is 0.125 to the power of number of recombinants. In this particular example, the recombinant that we found was 1 into 1 minus theta, that comes to be 0.875 to the power of number of non-recombinants. Here in the example, out of 8 progenies, 7 were non-recombinants, divided by half, that is 0.5 to the power of recombinants, that is power of 1, into 0.5 to the power of number of non-recombinants, that is 7. Here, after calculation, we get the value as log 10 12.566, which is equal to, once we find it from the logarithm table, LOD score comes out to be 1.099. Now, in this case, the LOD score is less than 2. Hence, it can be concluded that the gene for Neil-Patella syndrome and gene for blood type are not linked to each other. Now, in the third instance, as discussed in the earlier slide, if the recombinant frequency need to be calculated, then we need to find the values for recombinant frequency at varying levels, that is 0.001, zero point 0.1 till 0.5, and the correct theta value will yield the highest LOD score. I have given an example here. Now, first case, if we assume that the theta value is 0.125, that is the recombinant frequency is 0.125. Now, the probability that the first birth is the parental genotype, that is the first child is parental type, the probability is 1 minus theta, that is 1 minus 0.125, which is equal to 0.875. Now, when we learn the crosses, we know in the progeny we would get two parental types. So here each parental value would be 0.875 divided by 2. That comes to be 0.4375. Similarly, in a single crossover, in the progeny, we get two recombinant types. So here again, for recombinant type probability for each would be 0.125 divided by 2, which comes out to be 0.0625. What I'm talking here is the haplotype. In this particular example that we studied, there were 7 parental and 1 recombinant. So the probability of birth sequence at theta value 0.125 would be each parental value, that is 0.4375 to the power of 7, into the recombinant haplotype, that is 0.0625 to the power of 1, which comes to be 0.00019117. This is the probability of birth sequence if linkage happens at theta value 0.125. If there is no recombination, we take the value of theta as 0.5, because 50% above we consider as no linkage. Then the probability of birth sequence is 0.25 to the power of 8. That is, all the 8 individuals are parental types. There is no recombination. So here the value comes to be 0.000153. So the LOD is log of probability by linkage to probability by non-linkage. It comes to be log of 12.566. On finding the logarithm, the LOD score comes to be 1.099 at theta value 0.125. Similarly, I have found if the theta value is considered as 0.10, the LOD score would be 1.08677. In this, I have put the values that we got when we considered the recombination frequency at 0.1 and 0.125 in a table form, where the LOD score for 0.1 was calculated to be 1.08677 and 0.125 to be 1.09803. I would like you to fill the column, that is, calculate the LOD score for 0.05, 0.15, 0.2 and 0.25. Among the values, then identify the one which has the highest LOD score. That is considered as the recombinant frequency. In general, this is done by using certain softwares like MapMaker and LOD View. Here is an example of a case study wherein the researcher group could identify a new gene at chromosome 2p15 close to marker D2S393 and D2S3 S337 using pedigree analysis and LOD score. If you see the graph on your right, the markers at D2S393 and D2S337 show a LOD score above 3. The reference is given below. Thank you.